Would like to welcome our next speaker. This is Professor Pavel Levkin. He's going to give a talk on functional surfaces and micropatterns from superhydrophobicity to drug discovery. And Professor Pavel Levkin is a professor and head of department of the functional materials at Karlsruhe Institute of Technology, KIT, at the Institute of Biological and Chemical Systems. He's a co-founder of two spin-offs. The one is ScreenFact, the other one Aquarate. His group has published about 165 papers and submitted 20 patents. He actually received the ERC starting grant and 2 ERC proof of concepts for the EU and the Heisenberg professorship from the DFG. He also received a Heinz Meyer-Leibniz Prize and the Ewald Wicke Prize. And well, he also serves for the editorial advisory for several Wiley journals. And with this, I would like to give the floor to you. Thank you very much for being here today.
Thank you. Thank you for the nice introduction, and good morning, ladies and gentlemen. Thank you also for inviting me here. Second time, I guess. It's really nice to be here. I apologize from the beginning for my coughing because I haven't recovered completely from a cold. Today I will talk about functional surfaces and micropatterns from superhydrophobicity to drug discovery, but I want to focus on kind of the importance of functional surfaces, interfaces to accelerate drug discovery through miniaturization of biological and chemical experiments. And the best way to show the importance of miniaturization is by showing this plot, which is the famous Moore's law, which shows that number of transistors per microchip has been doubling every 2 years over the last 50 years. And this is basically miniaturization in combination with parallelization, throughput, and integration. This resulted in this drastic reduction of costs of microchips, which in turn led to the democratization of the technology of the microchips, personalized computers, which in turn resulted in the extremely positive impact on society, and from which all of us benefiting every day by using cell phones, computers, internet, cars, and so on. And the same should be true actually for life science, for biological experiments, for chemistry, for medicine, diagnostics, for drug discovery. And in fact, scientists have been working in this field to trying to miniaturize biology and chemical experiments. And on this graph you can see the number of papers increased from few hundreds of papers citing or mentioning lab on the chip and microfluidics in 2000 to tens of thousands of papers in this field per year nowadays. And in fact there are some success stories, and probably the most important success story is the drop of costs of genome sequencing, which dropped from hundreds of millions of US dollars in 2000 to less than $1,000 per sequencing this year. And I would argue this was only possible through these 4 things, through miniaturization, parallelization, high throughput, and integration of different parts of the process in the same technology, in the same system, which resulted in the reduction of costs first, then which led to the democratization of the technology, and we will see positive impact from this technology yet. And by seeing this, it's even more surprising than to see this plot, which is so-called Irum's law, which is opposite from Moore's law, which shows that the number of drugs that have been developed per 1 billion US dollars has been constantly decreasing over the last 70 years, which is, in other words, the costs for drug development have been constantly rising over the 70 years, despite of all of the miniaturization, lab on the chip, microfluidics technologies. And I would argue this is not actually despite miniaturization, this is because we actually failed to miniaturize biological experiments and chemical experiments. And here you can see the first attempt to miniaturize biology, which was done in 1950. This is a 96 well microtiter plate, which was introduced at that time, which was already a huge kind of success in terms of miniaturization. People went from Petri dishes and milliliters to 96 tiny Petri dishes and using just 200 microliters. But then the next kind of wave of miniaturization happened only 30 years later when the 384 well plate was introduced. And if you ask biologists nowadays, most of the biologists still use 384 well plates or even 96 well plates. So this doesn't really follow the famous Moore's law in life sciences. So there's basically zero miniaturization over the last 40 years. Of course, on average, there are good examples. But to please biologists, or probably also here, in chemistry, the situation is even worse. We still use the same hardware, glassware that people used 100 years ago. We still use milliliters, hundreds of milliliters for doing chemical reactions nowadays, and not even microliters, and I'm not talking about nanoliters. So I would claim that we can achieve the same kind of breakthroughs as in the case of semiconductor industry or in the sequencing, but we still have to further miniaturize biology to go beyond the limits of microtiter plates, to go to nanoliters, and at the same time we have to miniaturize chemistry to the same scale and the same throughput as biology, because it's essential to integrate chemistry with biology. It will not work if chemists still use hundreds of milliliters, and then finally biologists are trying to do millions of reactions and experiments in nanoliters. And this is what we have been trying to solve in some of the projects in my group by developing so-called open droplet microarray technology, which is basically a glass slide with immobilized thousands of nanoliter sized droplets in precise locations, where these droplets can be used for both biological as well as chemical experiments, but using 1,000 times smaller volumes. And this brings me, connects this important problem with the field of surface science, surface functionalization, patterning, because without this, it's not possible to solve this problem. So what is droplet microarrays and how do we make them? So in my lab, we have developed different methods to functionalize surfaces with, first of all, such superhydrophobic coatings, which are based on micro-nanostructuring and surface functionalization, which are extremely water repellent. And on the other hand, the opposite of superhydrophobicity is extremely hydrophilic or superhydrophilic coatings, which is also based on micro-nanostructuring and surface chemistry, but has completely opposite, highly adhesive properties. And then we also have been working on methods to create these surfaces using photochemistry based on UV lights. Here are some examples of photochemical methods that we have introduced. And the advantage of photochemistry or UV light is that we can control spatially where we can get the superhydrophilic spots and superhydrophobic backgrounds, because we can use a photomask and we can pattern surface with these two important properties. And if we use these methods to make an array of hydrophilic spots on a superhydrophobic background, then this leads to completely new, interesting functionalities, like shown here, the effect of discontinuous divetting, where if you just roll a droplet of water, you feel these thousands of compartments in precise locations with nanoliter droplets that can be used for individual experiments. And then again, by using controlling the geometry of hydrophilic patterns, it's possible to control shape of these droplets, size of these droplets, location on the surface, density, and so on. Then the new technology that has been developing over the last years for liquid dispensing also allows us now to precisely deposit controlled liquids and volumes of cell suspensions or reagents into precise locations, and we can control the volumes down to sub nanoliters. So the technology is there for these applications. With different liquid dispensers, as shown on the left-hand side, it's possible to print cells or single cells into these compartments, or bacteria cells, or other types of microorganisms and reagents. So with this technology, what we have been doing over the last few years, we have been demonstrating and optimizing biological applications, high throughput applications using nanoliter-sized droplets, which is 1,000 times smaller already than what people usually use in biology. So we have shown, for example, drug screens using cancer cell lines, using primary cells, which is important for personalized medicine, where there are not enough cells, where miniaturization is also important. For stem cell screening, single cell screenings, we recently screened several thousand compounds with bacteria to search for new antibiotics, and also showed the possibility of using this miniaturized high-throughput screenings of cells in three-dimensional microenvironments like hydrogels. But today I want to show you some examples of novel applications of droplet micros where which are not possible in microtiter plates because of the kind of design and the features of droplet microarrays. And the first one is adhesion versus gravity. Microtiter plates and the common biological and chemical experiments, they are based on gravity. So this is how we confine liquids in the well, so just use gravity. But if you reduce volumes to sub-microliters, then adhesive forces become much stronger than gravity, so you cannot actually use gravity. That's why you cannot miniaturize microtiter plates, basically. In our case, we actually use adhesive forces to control and confine and manipulate liquids at this scale, but at the same time, they become also much more stable on these droplets, on these droplet microarrays. And you can basically do biological experiments, cell experiments in any orientation of these slides, which you wouldn't do with microtiter plates, probably. And this is interesting because if you culture cells in the inverted orientation, you form cell spheroids in these droplets using the hanging droplet method. So cells organize into three-dimensional architectures in every single droplet, which you can see here, these dots, these are actually multicellular spheres. And this is important because cells in these 3D spheroids, they feel closer to the physiological conditions, and they respond to drugs also in a different, more physiological way. So then we used this method recently in a collaboration. Sorry, the colors are still missing. But we used it in a collaboration with Seven Turkan from Heidelberg, who is working on brain tumor, on glioma, which is aggressive brain tumor. And we used patient-derived and also drug-resistant glioma cells to form tumor spheroids on the droplet microarray and then screened them with 2,200 FDA-approved drugs for repurposing. And in total, it was 20,000 experiments with all the repetitions. It was only possible through miniaturization. But importantly, the screening resulted in 20 hits from these 2,000 compounds, and new compounds that were active against these drug-resistant glioma cells, and 3 hits were validated in vitro and in vivo, and also a new mechanism of action was shown, and hopefully this will lead at some time to new therapy against this bad disease. Another interesting difference of droplet micros from microtiter plates is that there are no walls around the droplets like in microtiter plates, like plastic walls that you cannot remove. And this means that you can merge neighboring droplets because there is no wall by liquid dispenser, and this brings, for example, cell spheroids together, which you can see on the left, into the same compartment. And then if we continue to culture these two spheroids in this compartment, they start to fuse, they start to interact with each other, and this leads to the formation of such a kind of giant cell spheroids, three-dimensional architectures. And here you can see, so we can merge 2 droplets or 3 droplets or 4 droplets, and this leads to three-dimensional multicellular constructs with very interesting architectures composed of multiple spheroids. And all of this is done in an automated and kind of high throughput way. What is also interesting here is that you can then culture different types of spheroids from different cells and then merge them, and they form also this type of complex 3D cell structures with interesting geometry, but also composed of different cell types. And what is also interesting here is that the fusion process doesn't lead to mixing of the cells. So we end up with such architectures where the boundaries between different cell types are still visible. And this is important to mimic natural like tissue architectures where you have different cell types, obviously, and interfaces between cell types. It's also important for making more physiological drug models for drug screenings. And here are some more images. So this is such a so-called cell assembloid from 2 spheroids with a clear boundary, from 3 spheroids. Again, you can see very nice boundaries, and even from 4 spheroids that leads to this formation of 3D giant spheroid with clear boundaries between different cell types. Another interesting difference of droplet micros from microtighter plates is the following, and it's based on the fact that in microtighter plates you have liquids that are confined in cavities or in wells like here, so they're inside of the cavities. In the case of droplet microarrays, the droplets are actually above the surface, and this makes them accessible for parallel manipulations using just another flat surface in a single step, like shown here. And this is unique because if you have like thousands of droplets, their manipulations or compartments have become very, very difficult. But since they are above the surface, they are addressable from the surface in parallel. And this can be used, for example, for copying a library or taking samples in parallel from all of these thousands of compartments, or for adding reagents in parallel to all of these compartments. And we used it recently to demonstrate in simple but important application for medium exchange, because if you culture cells for more than 3 days, you have to exchange the mediums, otherwise cells die. And how would you do this if you have like 10,000 of droplets of 200 nanoliters or compartments, and it's even hard to see them? How to exchange the mediums? Because usually biologists use pipetting robots and pipettes, but here it's not possible. But with this sandwiching method, it was possible to like remove part of the medium and then print fresh medium, and this resulted in on the right-hand side, you see it's without medium exchange, so cells start to die, and you have the last bar. So these are dead cells, these are viable cells. And with this medium exchange, you see increased proliferation and improved viability after 7 days of culturing of cells in 300 nanoliters, and all of this was done in parallel. Another interesting application of the sandwiching method is to do parallel addition of spheroids or transfer from one slide to another. And so here's a scheme. So if you do the sandwiching between 2 arrays containing individual cell spheroids, during this sandwiching step, when the bridges, liquid bridges are formed, spheroids, they kind of precipitate to the bottom slide, and when you open it, they will become trapped in the bottom droplets in parallel. And here you can see on this video, so initially there is just one spheroid, and then the sandwiching process, the second spheroid is added to the individual droplets in parallel. And this is another, like it's an image of droplet microwave with individual spheroids. So these dots, these are cell spheroids in every single droplet. And then after sandwiching, you see that each droplet contains now 2 cell spheroids. And what is kind of cool that you can repeat this process, and then you can add a third spheroid to the same droplets. And of course, you can then change spheroids and cell types and so on. So this opens a lot of possibilities for applications. But it's still kind of exciting because this is a single-step process where you can address literally thousands of compartments that are so small that are difficult to see. And then at the end, you see such a accuracy that every droplet has like precisely 3 spheroids. So it's kind of exciting. So now I come to the last part of my talk, which will be about miniaturization of chemical synthesis in these droplets. And this is, again, essential because if we don't do this, it will not be possible to integrate chemistry with biology. And this is important to achieve this integration step and to kind of try to realize the same promise as with semiconductor industry, with miniaturization, and with sequencing, because integration of components into the same system is essential. But how to do synthesis in 100 nanoliters, that's also not trivial. First of all, because usually chemists, and I'm a chemist, we do chemistry usually in organic solvents. Only green chemists probably work with water. And organic solvents, they have low surface tension, and low surface tension is not good for superhydrophobicity, because superhydrophobic surfaces are also known as oleophilic. And to confine organic solvents with low surface tension, the surface, again, it comes to the importance of surface functionalization, surface chemistry, but knowing how to functionalize surfaces and change their physical properties, it was possible to to make a different type of patterns that enables to confine even hexane with very low surface tension in precise droplets, but also other organic solvents. And I want to show you one example of an early study where we wanted to demonstrate all the advantages and to go from kind of drug discovery, go from organic synthesis, library synthesis on chip, to characterization on chip, and to biological screening using this exactly the same platform. Sorry. And for this we designed a chemical synthesis to synthesize this type of lipids, cationic lipids, using 3-component reactions using amine, thiolactone, and pyridyl disulfide that react together to form a cationic lipid, and we can then use different building blocks to make a library of lipids with different structures. And cationic lipids are important because they are the main component of vaccines, for example, mRNA-based vaccines, corona vaccines, or cancer vaccines that have been developed by pharma companies. And the right structure, performance, toxicity, activity of this cationic lipids is essential for the next generation vaccines. So the synthesis then was done on the droplet microwave by basically adding all 3 components for the chemical reaction into the individual droplets using also sandwiching method and the discontinuous dividing method in this case. It was done in DMSO, then reaction happened overnight in DMSO, and then we had at the end droplets, each droplet containing a different type of lipid. Then these lipids were also characterized basically on the same platform, and using the advantage of droplet microwave that we can stamp it basically on a multi-plate, which is used for multi-mass spectrometry analysis, and we could analyze all of the lipids then in parallel on the same slide. In the next step, we took this library of lipids and then sandwiched it with another slide containing aqueous buffers with plasmid DNA to form lipoplexes or lipid nanoparticles. So lipid nanoparticles complex with nucleic acids. And at the end, we basically got a library of LNPs formed from different types of lipids in this on this array. And the last step was basically just printing cells to this LNPs on the surface and then incubating this droplet microwave for 1 or 2 days and then analyzing by fluorescence microscopy the number of transfected cells that express GFP and comparing to a control. And this screening already resulted in some lipids that were more efficient for cell transfection than commercial control. But what is essential here, I think what I'm always trying to emphasize, that in this early study we did on-chip synthesis of 75 lipids, on-chip characterization of all of these lipids by MALDI, on-chip lipoplex formation, on-chip screening with cells. And all of this was done within just few days at the end and just using 1 milliliter of solvent. And this is, I think it's a good example how miniaturization, parallelization, high throughput, and integration is important and can actually lead to drastic reduction of costs for example, early drug discovery, and eventually should lead to democratization of this technology and positive impact on society. And with that, I would like to thank all of my team members who contributed to this work, present and also alumni, our funding funders, and I also want to use this opportunity to congratulate you guys for these 3 wonderful journals, and thank you for your attention.
Well, thank you for this wonderful talk, and with this, the floor is open for questions. Over there.
Thank you for this fascinating talk. I was just wondering, when you talk about very small droplets, to which extent actually volatility of the solvents play a role?
Yeah, that's important. Of course, the evaporation of liquids, I'm sorry, becomes an issue. But when we go to really small volumes, then we have to control the environment and the temperature, humidity. But this is solvable. So, let's say it's not the big issue. It cannot be done in the same way as microtiter plate. We just open it and kind of walk around. So it has to be done in a closed environment, and students have to work quickly. But for water, is down to 200 nanoliters is not a problem. Yeah, you have to be skilled, but going below 200 nanoliters, then it would have to be done in a controlled atmosphere, and then it still works. For organic synthesis, usually we have to work with, in this case, with low volatile solvents like DMSO, DMF, and NMPs, which work for most of the chemistries. Or we have to, again, do it in a controlled environment, yeah.
Thank you for the beautiful talk. I wanted to ask how you control which side the cells go to when you do the sandwiching and then you pull the plates apart. Can you control to do successive operations?
How can we control
which side the cells stay on after if you separate the plates?
In this case, so if you talk about the sandwiching of
this one.
spheroids, that's a good question. So this is actually very simple. Cell spheroids, they're relatively big, and once we have this sandwiching step, which you can probably see better here. Yeah, so there is a liquid bridge, so the two droplets that are connected, and spheroids, they basically precipitate to the bottom slide. When you open it, they're all trapped at the bottom slide. So you can actually, if you invert it, you go to the other side. You can transfer it to the other slide as well, yeah. Yeah.
So I wonder how the effectiveness of this technique for when it goes to in vivo. I mean, you screen some drugs, then you probably have to test it in vivo. And how, I mean, as a consideration on the real, like, conditioning, comparing with in vivo testing.
So if we talk about early drug discovery, where the first step is the screening, usually huge libraries of compounds with cells or proteins to find binders, and then you identify hits. Then they have to be further improved. They have to transfer to lead. Structure should be improved, and this is done in parallel with an in vivo applications. But this is the next step, the preclinical tests, and then it's going to clinics. So the first step is always in vitro, and the chemical space is enormous, and even pharma companies that have millions of compounds, they're still covering just like tiny portion of compounds. Of course, there are other methods, DNA tagging and so on. But still, it's important to increase the chemical space that is accessible to all these biological models to find initial hits, and then they will be improved and they will go to in vivo. But we don't do in vivo in this step, so focus on the first step.
I think we have to continue with the schedule. I'm sorry. I actually also have a question. I will find you later to ask it. So thank you again for this really nice talk.
Thank you.